Hi guys, this time around I will take the opportunity to provide a very quick look into microbial life, in particular to a very specific subject, the bacterium. As you may have had a chance of taking a look at under my blog at creationfactory.blogspot.pt, I have recently scored from eBay a 50-year-old Olympus microscope, which in spite of my modest expectations, I found to be in pristine condition. Since the moment I unpacked it, I have been doing several experiments and observations into the microbial world. I have not hesitated in adding a proper converter to fit my SLR and starting to shoot video and taking photos. For instance, in my previous video, you may have seen some observations of several types of microorganisms such as the protozoa, the algae, and bacteria. This time I decided to run another typically academic experiment of attempting to culture bacteria in a Petri dish filled with nutrient agar. I took these Petri dishes, which I had previously sterilized with ethylic alcohol. I filled these recipients, these dishes, with nutrient agar, and after cooling down and solidifying, I rubbed each one with a cotton swab contaminated with bacteria from exposed surfaces, namely from a computer mouse, from the outside of a jar containing the Paramecia culture that I've used for shooting the previous video, and also a used car part, namely a piece of the air intake system. With the one contaminated from the computer mouse, I let the culture develop in a close, darkened environment for a few days, until signs of culture development became visible to the naked eye. Then I prepared a microscope and the camera for long-term shooting of periodic photos to create a time-lapse sequence, a time-lapse video that you will see next. It took roughly 24 hours to create these images. Each circular culture normally derives from a single original bacterium, which divides further on, giving place to a culture. This serves to show the formidable ease with which bacteria are capable of multiplying in what are close to ideal conditions, as is the case of the nutrient agar. One aspect that is clearly visible and for which there is likely a commonly accepted explanation is that up to a certain point, adjacent colonies seem to repel each other and form a gap up to a certain point. However, as colonies continue to grow, this gap eventually closes because there is really just no more space where cells can grow. But still, a line remains visible. If you search, there is a book called The Physical Basis of Bacterial Quorum Communication by Stephen J. Hagen, which also describes this phenomena. And on the study that follows, the author relates this occurrence with the SLF toxin that is produced by the bacteria when their density increases in a confined space. You can follow the link added down below to find more information about this study. In the surfaces that we come to contact with in our everyday life, we normally don't expect to be exposed to such high concentrations of bacteria as it happens to be in a Petri dish like this. We should not forget, however, that if given the chance, bacteria will reproduce in a similar fashion to a culture like this. And with higher temperatures, there is a greater chance of pathogens reproducing and spreading. One such example is the Legionella Bacteria, for which the colony's development and spreading is always a risk, in particular in environments that involve humidity, also exposure to the outside atmosphere, and whenever the range of temperatures is similar to the human body core temperature. Large industrial settings containing water cooling towers, when these are not properly and regularly inspected, constitute a risk for the general population. So guys, I hope you enjoyed this video. I hope you had a good time while watching this. I hope you could learn just as much as I did by doing these observations, by trying these experiments, growing cultures of bacteria, of algae, of protozoa. Even though for me this is a rather new topic, I'm not a biologist by profession. I am a computer software engineer, but well, I have curiosity in other topics, which enable me to, well, try to dive a little bit into these areas as well, and learn and share my learning process. For a long time, I didn't had contact with microscopes since the times of high school. It was an interesting journey to you know to do this stuff, since from the process of restoring this old microscope, as I had the chance of writing in my blog, and you guys may again look at it and see, to actually preparing slides and looking at this wonderful world that is totally invisible to our eyes, but it's there, and it's very real as the world in our scale, obviously. So stay tuned, subscribe my channel, and keep watching these videos. Bear in mind that my channel is pretty wide in terms of scope. You may find not just biology videos, but also technology videos. I try to bring a multitude of scientific and technical areas, and again, like I said, share with you guys the same learning process that I've been through. Hoping to be able to stimulate and bring a little bit of that enthusiasm, the same that I had doing this stuff. Thank you. Bye.